My name's Terry Moody, and I work for the NCI Center for Cancer Research, and this is the course on redox biology. So we've done this course for about 10 years. We don't do it every year, every other year. And I'd like to acknowledge Jonathan Wiest of the NCI. He's the guy who approved the course and pays for all the video casts. And we'll have David Wink talk as the second speaker today. He's an expert on nitric oxide in cancer. So we have several courses at NCI. One which we're doing right now on Mondays is the Translational Research in Clinical Oncology, TRACO. And on Tuesdays now, we're doing the redox biology. And then in the winter and spring, we do additional courses such as statistical analysis of research data, cancer biotechnology, and others. And the advantage of NCI courses is the price is right. It's free. And so we have 2 lectures each Tuesday. And I'll be doing the introduction today. Dave Wink will be talking about the chemistry. Then next Monday, I'll be talking about signal transduction. Dave Roberts will be talking about angiogenesis. So for nitric oxide, especially, the big thing that's produced is cyclic GMP. That's a signal. And with cancer, as tumors grow big, they need to be infiltrated by host blood vessels so that the tumors can get glucose and oxygen and continue to grow. And then Dave will be talking about the biology on October 10th, and Stefan Ames will be talking about epidemiology. And then on October 17th, Feroz Hussein will be talking about inflammation. About 20% of the cancers result from inflammation. Tom Lado will be talking about NADPH oxidases. On October 12th, 24th, Lisa Ridner will be talking about enzymes. Paul Patcher will be talking about redox physiology. On October 31st, Dave McVicker will be talking about immunology. Dr. Bad Suhar will be talking about HO and nitric oxide. And then we'll be closing with Murali Krishna talking about imaging. And we have one additional lecture yet to schedule. So as we mentioned, the course is open to all interested NIH personnel. There's no charge. And then an advantage of the course is it's video conference to NCI Frederick. NCI is a very big institution, and about 1/4 of our staff are up in Frederick. And then while we videocast it live, it's also archived as a past event. So 2 days after the lecture, then you could call it up on your computer at videocast.nih.gov. You can watch it anytime, and it stays posted forever. And then you can obtain a course certificate upon passing a final examination. So it's computer graded, multiple guess questions, and it will be posted at our website, which is ccr.cancer.gov/trainees/resources/courses/workshops/RB. And also we'll be posting the PowerPoints at this website as well. So are there any questions on the organization? Okay, then we'll proceed to the first lecture. And so I'll be introducing you to the various gaseous species that are biologically active. We have reactive oxygen species, such as hydroxyl radical. We have reactive nitrogen species, such as nitric oxide. And additional gases that we'll be discussing today include hydrogen sulfide, carbon monoxide, and hydrogen. So we'll start with the reactive oxygen species. And so one of the key species is the superoxide, O2 minus. So superoxide, it's especially released by immune cells to kill pathogens in the body. And then another reactive oxygen species is the hydroxyl radical. Another is hydrogen peroxide. The first two are gases. Hydrogen peroxide is actually a liquid. We use it in experiments in the lab all the time. It's an oxidizing agent. It can also be used as a bleaching detergent and a disinfectant. And then another gas is ozone. And I personally experienced this when I was a graduate student at Caltech in Pasadena, California. Los Angeles has lots of air pollution. It starts out in the main part of town, but then as the day progresses, the winds blow it east, and it gets to Pasadena about 1 p.m. You're in your lab working, and suddenly your lungs start burning, and the oxygen, it just causes pain. And then about 3 o'clock, it would all blow further east to Riverside, and you could breathe more normally again. Another reactive species is the singlet oxygen. And then what happens is when you get exposed to this oxidative stress, a lot of your lipids undergo peroxidation, and your cellular membranes can become brittle, and then cells can die. When I was a grad student at Caltech, I was told for every 2 years you stay here at graduate school, you're going to lose 1 year of your life just to all the oxidative stress that you're undergoing from the bad air. In contrast, the DC air is much better, very little ozone. And so these reactive oxygen species, they don't just act one by one, they act as a group. And so we see here we have superoxide. It can then form hydrogen peroxide, and then it can form hydroxyl radicals. And these reactive oxygen species, they can oxidize DNA. They can oxidize lipids, as we mentioned, and they can also oxidize proteins, leading to cytotoxicity. So we have enzymes in our body then that can protect us from this enzymatic stress, one of which is superoxide dismutase, and it protects us from the superoxide. So the superoxide reacts with hydrogen to form O2 and hydrogen peroxide. So superoxide dismutase, it's in the cytosol, it's present as a homodimer, and it's a relatively small protein, just 153 amino acids, a molecular weight of about 16 kilodaltons. Catalase protects us against hydrogen peroxide toxicity. So we see the hydrogen peroxide reacts with oxygen, reacts, and it goes to form oxygen then and water as the products. So catalase, it's a homotetramer, which is localized to the peroxisome, and it's got 526 amino acids, a molecular weight of about 60 kilodaltons. Glutathione peroxidase also protects us against hydrogen peroxide, and glutathione reacts with the hydrogen peroxide, and the glutathione with a cysteine gets oxidized to a glutathione with a disulfide, and water is also a product. So glutathione peroxidase 1, it's a homotetramer in the cytosol. It's got 201 amino acids, about 22 kilodaltons. And an important enzyme is the peroxyredoxins. These reduce hydrogen peroxide, organic fatty acids, and phospholipid hydroperoxides. And we have cysteines interacting with the peroxide, going to form a disulfide, and an alcohol is released as the product. So pyridoxine 6, it's a homotetramer present in many organelles, and it contains 223 amino acids, weighs about 25 kilodaltons. And oxidants, they can affect two main pathways. At low doses, they can activate MAP kinase, leading to cellular proliferation. And also NF-kappa B can be affected. This causes the release of cytokines and alters the cell cycle control. There's Dave. Sorry. He works up in Frederick, so he had to drive down 270. We're lucky he's here. Okay. And so reactive oxygen species, they're involved in heart disease and especially aging. Your body is constantly exposed to oxidative stress, and Alzheimer's disease can certainly uh, be facilitated by reactive oxygen species. And our focus is going to be cancer. And so since these oxidants are basically bad, uh, the question is, will vitamins have an effect on patients? Because vitamins have lots of reductants. So in general, in cancer, you have a chemical, it gets oxidized to a carcinogen, and this starts the carcinogenic process. And it can take decades before a patient will get cancer. So these studies have been done, especially using vitamin C, vitamin E, and vitamin A derivatives. And basically, these did not stop the cancer. And so in France, there was one study that was done with 13,000 men and women, and half of them were given antioxidant vitamins while the others received a placebo pill. And the vitamins included beta carotene, vitamin C, vitamin E, some selenium, and some zinc. And basically, there was no difference. So vitamins can make a difference if you're vitamin deficient, but most people in the US have plenty of vitamins. Now we're going to look at the reactive nitrogen species. And so we first go to Dave's favorite, which is the nitric oxide. We have enzymes, the nitric oxide synthase, that can metabolize arginine to citrulline and nitric oxide. We have three forms of this, the neuronal form, the endothelial form, and the form that's in the cancer cells is the INOS. This is an inducible enzyme. And so here we see the arginine being exposed to oxygen. And then we have the enzyme, an intermediate is the N-hydroxy L-arginine. And then with further oxidation, we get citrulline as a product, which is in the citric acid cycle, and the nitric oxide. So nitric oxide in general, it's a very short-lived species. Okay, so the nitric oxide then, it can react with superoxide, giving reactive nitrogen species. And one of the species is nitrogen dioxide, and we mentioned about all the air pollution in LA. Well, the air there, it's orange, and it's orange because of this nitrogen dioxide. And sometimes in the summer here in DC, we see that the sky sort of turns hazy and orange in color, but I assure you, that's nothing compared to what happens in LA. And then we have other species, such as the dinitrogen tetroxide and the NO2. Two of these can react to form the N2O4. And then other species include the nitrite. Nitrites are generally used in this country as a food preservative. And so if you go to the store and you look at the beef, you'll see it's all red in color. And the reason for this is the grocer has put nitrite on your meat, and it reacts with the myoglobin that's in the blood vessels of the meat, and it turns it red. And if you wait for a day or two, you'll see it turns brown. That's because all the nitrite has gone away. So when I eat my meat from the store, I wash it first to get rid of the nitrite. We also have nitrate. This is used in fertilizer. And unfortunately now we have many terrorists and they've learned to use this in explosive bombs. So nitrate you have to be very careful of. And then we have peroxynitrite. So this can form when nitric oxide reacts with the superoxide. And so one of the things about cancer cells is we have this enzyme, prolyl hydroxylase. And I forgot and left this out of your handout, so you have one page with this diagram of it. And you'll see the prolyl hydroxylase, what it can do is it can react with a protein called HIF1 alpha. And it can hydroxylate it, and when it does this, then the H1F alpha binds to the Hippo von Landau protein, VHL. It gets polyubiquinated and then degraded in the proteasome. But reactive nitrogen species as well as reactive oxygen species inhibit this prolyl hydroxylase. So then the HIF1 alpha doesn't get degraded, and then it can go into the nucleus and alter gene expression. So as HIF1 alpha accumulates, it can form a heterodimer then with HIF1 beta in the nucleus of the cell, and the P300 CBP binds to the HIF1 alpha complex, altering gene expression. So another thing that happens is the reactive nitrogen species especially cause the P53 expression, as does hypoxia. And then the P53, it gets phosphorylated as a result of reactive nitrogen species and hypoxia, affecting the ATR, ATM. And then when it gets phosphorylated, it goes into the nucleus and alters gene expression. And alternatively, when it's not phosphorylated, such as then the P53 can get degraded by the proteasome after it interacts with this MDM2. So this is all occurring in cancer cells especially. And HIF1 alpha, it basically helps normal tissues and tumors survive hypoxic conditions. And so in cancer, this is a big deal because initially you have cancer cells, these form small tumors, but then as the tumors get bigger, it's hard to get oxygen into the center of the tumor. And so this HIF1 alpha helps then get the cancer cells to survive the hypoxic conditions. And HIF1 alpha, it alters over 40 genes. We have group 1 genes such as transferrin and VEGF, which is an angiogenic factor, deliver O2 to the tumor. It affects group 2 genes such as enolase and hexokinase to provide sugars to generate energy so the tumor can grow. And then group 3 genes such as IGF-2 and IGF binding proteins 1 and 3 increase proliferation and viability of the cancer cells. So then reactive nitrogen species, it causes phosphorylation of the P53 at serine 15, and then it recruits the coactivator P300 CBP to alter gene expression. And so some of the genes that are affected are the hexokinase, which is the group 2 gene, the EGF, VEGF, which is a group 1 gene, and IGF-2, which is a group 3 gene. So this illustrates the process of carcinogenesis. We have normal cells, but then there are mutations in the genome, especially this is illustrating colon cancer. So the APC gene and beta-catenin get mutated. And then further mutations include BRAF, TGF-beta receptor 2, SMAD2, SMAD4. The tumor gets bigger. And then finally, P53 gets mutated. And when this happens, then the cancer can undergo metastasis from the primary organ, the colon, to other organs, such as the lung. And so for cancer cells, we require angiogenesis to give the tumor oxygen and glucose, and then metastasis to distant organs. And the angiogenesis starts when the tumor is 1 millimeter in size, and the metastasis occurs when the tumor is larger, about 5 millimeters, and the cancer can then undergo metastasis to distant organs through either blood vessels or the lymphatic system. And so this illustrates the process of hypoxia. As the tumor grows larger, you get further and further from the blood vessel, so the oxygen levels decrease. And this illustrates the normal breast. You have lots of small vessels in the normal breast, so the oxygen levels are fairly high. But for the tumor, they're high right adjacent to the vessel, and then they drop off dramatically. And this comes into play then in terms of transcriptional activity. The AP1 gene is activated when cancer cells are exposed to reduced oxygen, and the NF-kappa B pathway is also activated then when cancers are re-exposed to normal oxygen pressure. And if you're treating a patient with radiation, you see if the oxygen levels are very low in the tumor, the radiation isn't very effective and the patient dies. This is what's called a survival curve. In this case, half the patients are dead after about 25 months. If you give the radiation in the presence of oxygen, it's more effective, and the patients, about 25% of them have died after 5 years. So this is why we do radiation therapy. It increases the survival of patients, especially if the tumors have lots of oxygen. And now we're going to go to our third gas, which is hydrogen sulfide. So the reactive oxygen species, reactive nitrogen species, they're the most important, and the most is known about these. Less is known about H2S as well as carbon monoxide. And H2S, it's known to inhibit caspase cleavage and MAP kinase phosphorylation. So it promotes the survival of cells. And the sulfur amino acids are methionine, which is an essential amino acid. And here we see the sulfur of the R group. And it's metabolized into cysteine, which also has a sulfur. And these are the reactions that occur. Methionine is an essential amino acid in your diet. It's metabolized by methionine synthase to serine and homocysteine. Homocysteine is then metabolized by cystathionine beta synthase, CBS, to cystathionine. Cystathionine is then metabolized by gamma cystathionase, CSC, to cysteine. And we note here then that as a result of the CSE, H2S is released. Cysteine can then be metabolized by cystathine aminotransferase to mercaptopyruvate. Mercaptopyruvate is then metabolized by mercaptopyruvate sulfur transferase to pyruvate. And again, H2S is released. So I used to teach protein chemistry to medical students. So I know all about these amino acid metabolisms. I've done this hundreds of times, this cycle. And the two big ones really are the CBS and the CSE. So there's lots of inhibitors of these enzymes, and also they've done studies using cells where these enzymes are knocked out. And so the CSE, these knockout mice have reduced serum H2S. They have cardiovascular dysfunction and hypertension. The CBS is also important. Loss in humans leads to homocystinuria, which affects the cardiovascular, skeletal, and ocular system, as well as the CNS. So it's these enzymes then that are the key thing in causing the hydrogen sulfide release. And hydrogen sulfide at low concentrations, it stimulates oxygen consumption, but at high concentrations, it inhibits the mitochondrial respiratory chain and cytochrome oxidase. And we mentioned about the cysteine being important. Well, in our body, we have a protein called glutathione. It's composed of 3 amino acids, glutamate, cysteine, and glycine. And so we see the cysteine is in the middle, the glutamate is at the N-terminal, gamma-glutamyl, and the glycine is at the carboxy terminal. And so this tripeptide then is a natural reductant in our body. And cancer cells are loaded with glutathione. So glutathione helps maintain various entities in the reduced state, including the iron, which Dave will talk about. The iron is in the myoglobin and the hemoglobin. So glutathione, it's the most abundant protein thiol in the cell. It's an antioxidant that provides cellular protection. It maintains the redox environment of the cell, and it's increased when the cells are stressed and downregulated after a challenge has been faced. And so this now sort of integrates some of the various processes that we've mentioned. So here we have superoxide being produced, and this illustrates the mitochondria. And we have cytochrome C in the mitochondrial membrane. And one of the key things here is we need lots of NADPH in the cell because that helps keep the glutathione reduced. So the reduced glutathione is GSH. The oxidized glutathione is GSSG. We see here in the cell we're producing superoxide. It's then being metabolized by the superoxide dismutase to the hydrogen peroxide, and then the glutathione peroxidase enzyme is reducing the hydrogen peroxide to water, and in the process, the GSH gets oxidized to GSSH. Then we can, when we've eliminated then the superoxide threat, we can then reduce the GSSH by glutathione reductase using the NADPH. So this illustrates how the cell puts these things together. And this is the biology of the redox in the cell. So another gas that we're going to talk about is the carbon monoxide, and it likes to bind to the iron in the heme. And in hemoglobin, actually the carbon monoxide binds tighter to the iron than does oxygen. The whole point of hemoglobin is it carries oxygen that you breathe in through your lungs to cells throughout your body. So the problem is then this carbon monoxide gas. And when it's released, it can lead to asphyxiation. So if you want to commit suicide, one way of doing it is you go in the garage, you start your car engine, you keep the car engine running. After about 10 to 20 minutes, you'll have a sufficient CO so that you pass out. And if you continue to breathe in the CO, you will die then because you won't have any oxygen getting to the tissues in your body. And so the carbon monoxide also inhibits the cytochrome C oxidase, and the carbon monoxide is released when hemoglobin is degraded. So the carbon monoxide, it can inhibit cytochrome C oxidase, and it also affects the MAP kinase phosphorylation. So carbon monoxide levels then can be reduced using enzyme inhibitors, the heme oxidase inhibitors. So the heme is oxidized to biliverdin and CO by the heme oxygenases. And the inhibitors include imidazoles and metalloporphyrins. So for the neuroscientists, we've added a little section here on hydrogen gas. And we don't have any enzymes in our body to produce hydrogen gas, whereas we have enzymes to produce nitric oxide, CO, and H2S. But the hydrogen gas, what it can do is it reduces the hydroxyl radical and the peroxynitrite in the cells. It's anti-inflammatory, anti-apoptotic, anti-allergic. It stimulates energy metabolism. So it's not toxic. It's not going to kill cancer cells, but what it will do is get rid of excess reactive oxygen species, such as the hydroxyl radical and the peroxynitrite. So the H2 gas, it readily crosses the plasma membrane, accumulating in the cytosol, the nucleus, and the mitochondria. It reduces excessive oxidative stress, but does not disturb redox homeostasis. And you can infuse this gas into water and have animals drink it. And you can then measure the hydrogen that's actually in the blood. And the whole key to this is the hydrogen gas in the water has to be stored in an aluminum container, not plastic or glass. And so chronic oxidative stress can result in neurodegenerative diseases, such as Parkinson's disease. And if you give the hydrogen water to rats after exposure to 6-hydroxydopamine, the incidence of the Parkinson-like syndromes are greatly reduced. So the hydrogen water decreases the loss of rat dopamine neurons in a nigrostriatal pathway and may improve mental disorders. So hydrogen gas is sort of a player of the future. It's being researched quite heavily in Japan, not so much in the US, but it's been shown to be useful in an animal model of type 2 diabetes. And it doesn't alter food or water consumption in the animals, but decreases fat and body weights. And hydrogen water decreases plasma glucose, insulin, and triglyceride by stimulating energy metabolism. So it's proven quite useful then in animal models. So we're going to do a summary now comparing the reactive oxygen species to the nitrogen species, the reactive nitrogen species to the hydrogen sulfide and the carbon monoxide. So I found a new review, and it calls these chemicals gasotransmitters. So nitric oxide, as we've seen, can be synthesized by the nitric oxide synthase, carbon monoxide from the heme oxygenase, H2S from especially the CBE. And the difference is nitric oxide is a diffusible and labile free radical gas that has a short half-life on the order of seconds. Carbon monoxide is diffusible in a labile gas that has a longer half-life on the order of minutes. H2S is diffusible in a labile gas that has a medium half-life in between the nitric oxide and the carbon monoxide. And the nitric oxide can react with iron heme centers. It reacts with protein cysteines to initiate S-nitrosylation. And it reacts with oxygen-free radicals, such as superoxide to form peroxynitrite. CO binds to the iron heme centers, such as hemoglobin, and it can then form carboxyhemoglobin. And H2S binds to protein cysteines to initiate sulfhydration. So NO, it activates guanylate cyclase, leading to an increase in cyclic GMP. It also opens potassium channels. Carbon monoxide weakly activates guanylyl cyclase, but it also opens calcium channels. And H2S actually inhibits cyclic GMP and cyclic AMP phosphodiesterase, but it also opens calcium channels in cells. And the nitric oxide, it inhibits cytochrome C oxidase, reducing cellular energetics. After conversion to peroxynitrate, it increases poly-ADP ribose polymerase activity, impairing cellular bioenergetics. CO inhibits cytochrome C oxidase, reducing cellular energetics. But H2S is different. It donates electrons to the mitochondrial complex 2 to stimulate bioenergetics. NO is an antioxidant at low concentration, stimulating cancer growth. NO can form peroxynitrite at high concentrations and is cytotoxic. Carbon monoxide activates MAP kinase at low concentrations, leading to proliferation, but is cytotoxic at high concentrations by impairing bioenergetics. H2S is an antioxidant at low concentrations, stimulating proliferation, but is cytotoxic at high concentrations. And then these are some references. There's one on the hydrogen and one reference on the gasotransmitters. And that's about it for me. Dave always wants me to finish up quick. Any questions? Okay, well, we sort of laid the foundation now, and we've discussed the nitric oxide and the reactive oxygen species and the H2S and the CO. And Dave's a real expert on the nitric oxide, and so he'll be presenting that next. So we have one announcement, and that being on September 30th, we end the fiscal year. And Congress has not approved us any more money as of yet, but we hope that they'll do it late Friday and that we can continue to function and teach the course. But I think it'll be okay because this is an election year. I don't think they want to close down the government this year. So Dave got a Ph.D. in chemistry at the University of California, Santa Barbara. Then he was a National Research Service Award recipient at MIT. He came to NCI in 1995, and he's been here ever since. He's big now in the Cancer and Inflammation program, and he's chaired a Gordon Conference in Italy in 2009, and he's also been a fellow of the American Association of Advancement in Science and Chemistry. And his title today is the Redox Biology Chemistry. Dave.
Thank you, Terry. Yeah, no, it's, I'm glad everybody came out. Every time we give this course, I always get since we founded the course, he said, I have to teach the freshman chemistry part. Remember that part that you went at 8 o'clock in the morning and you really hated? Okay, I didn't hate it so much, but I really didn't like it. Many people don't. But today what I want to do is pick up what Terry says, and I want to we'll talk a little bit more about the chemistry. So when you just simply read oxidation, what does that really mean? And it's kind of like saying light. How many colors of light are there? And blue is different than red. So the same is in redox. There are a lot of different reactions, and Mother Nature is much smarter than we are. And as we've taught this course over the last 10 years, we're actually starting to appreciate how the basic fundamental redox chemistry plays as an important role as phosphorylation, simulation, or any of the other different types of signal transduction. And that's why this is a ubiquitous system, because it actually transmits in real time the status of the cell. So what we're going to do is kind of take you through some of the chemistry today. Let's introduce our molecules. Terry's already done this, so I can go quickly through. You have the reactive oxygen species. These by themselves are fairly unreactive. Hydrogen peroxide, we can put it in a bottle, right? You go in the lab or you go home, right? I use it at home, get a cut, you put it on there. So it's fairly stable. But in the presence of metals, they undergo what we'll discuss later. It's called the Fenton reaction, and you generate these reactive species. Some of them are so powerful, they're like the same equivalents to that in radiation. We, of course, the nitric oxide species, my favorite, nitric oxide and all its progenitors. And also recently, I don't know if I'll have much time to talk about this, but HNO we started our investigation on HNO, and it turns out we and others have shown that we think this is actually as important molecule as where they gave the Nobel Prize in nitric oxide, that HNO is a very important endogenously generated species, and we've actually have a patent on HNO for heart failure, and it's now going to phase 3 clinical trials. Bristol Myers Squibb has actually invested heavily in this, and from what I understand, it's actually going to be the state of care for heart failure now. Carbon monoxide. A lot of neat chemistry about carbon monoxide. One thing about carbon monoxide is they actually use this in breathing this to overcome things like sepsis. So there's a very powerful effect on the immune system. So let's just talk about the basic chemistry. And then if you guys really hate that, you can go to sleep or leave. You know, I won't be insulted. You don't get graded on this, so it's okay. So basically, the way I look at this chemically is we have these instigators. These are fairly stable molecules. Remember, nitric oxide, though it goes away fast in the body, you can still buy a cylinder of it. It's really fun, actually. And then I was always a chemist, so we like to blow things up and see color changes, that kind of thing. So, but anyway, hydrogen peroxide. But when we activate the hydrogen peroxide, we generate our primary reactive species. This would be hydroxyl radical or iron oxyl species, which then can further oxidize lipids, proteins, other biomolecules, resulting in biochemical products like oxidized lipids, DNA. And then the ultimate thing is they form an oxidative stress in the biological system. Same thing with NO. NO by itself, we'll talk about that, has its own unique properties, but it can be converted in the presence of oxygen or ROS to these species here, which can result in nitrosamines, some of which are carcinogenic, S-nitrosothiols, which actually have a lot of signaling potential, nitrotyrosine, a footprint, and ultimately we can result in what we like to call nitrosative stress in the biological term. So in the redox species, the vast majority of stresses are either oxidative and nitrosative. And chemically, but also biologically, oxidative and nitrosative stress seem to be orthogonal. I don't know if everybody remembers that term, orthogonal. Either it's here or it's here, XY axis. One cannot happen in the presence of the other. So the redox stress concept. If you read in the 1970s and 1980s, you would think that all ROS is bad. People would say, oh, we're constantly getting irradiated because we breathe, we convert oxygen to peroxide. Peroxide generates hydroxyl radical. That's just like in radiation. We're all dying, and this is why we're dying. Well, that's not quite true. You may have chemistry that looks like an atomic bomb, but if you look at redox chemistry, it actually looks more like the phone box in the building. You kind of look at it, it seems infinitely complicated, but there actually is a reason, rhyme, and that's why we can just pick up the phone and call somebody. So there's a difference between stress and redox homeostasis. You cannot function today. We'll talk about your blood vessel. Nitric oxide relaxes your muscle. ROS contracts the vessels. You have this yin and yang happening all the time, and they're very critical in just regulating your day-to-day function. So what are the different mechanisms? Well, we have the chemical effects, like just straight up direct oxidation of lipids, metal-catalyzed DNA oxidation. We get products of oxidation like cholesterol, your oxidized LDLs. DNA base pairs can be oxidized and mismatched, resulting in spontaneous mutations. And also the biological redox balance. One of my favorites is ischemia reperfusion injury, where you block off the blood vessel. You create hypoxia. But when you open that blood vessel back up, all that hypoxia forms these reducing agents. And those reducing agents go right to oxygen. And instead of burning it slowly through the mitochondria and getting any DH out of it, instead what you do is you actually convert it immediately into hydrogen peroxide, which then signals a stress response. And it turns out that's critical in the ischemia reperfusion, recruiting leukocytes. And why do we want leukocytes to go to a damaged area? Because we want them to clean it up and make it antiseptic. So it's a very interesting kind of system. Shear stress in the coronary circulation is this balance between ROS and nitric oxide. So the fundamental concepts of electron transfer and oxidative chemistry. Boy, this is when you can go to sleep. Huh? I understand. You can go to sleep at this point. The chemistry and formation of reactive species, the DNA, protein, lipid chemistry, and then I'll talk a little bit more in general how all these reactions really apply in a biological system. We coined the phrase the chemical biology of NO, which is actually balancing things like nitric oxide and ROS. So I'm sure you haven't seen this since freshman chemistry, the Nernst equation. Ouch. Basically, we won't go through all this. I just want you to just get the idea that there are actually quantitative terms that we can actually put in context with how powerful an oxidant is and how powerful a reductant is. And very simply, this is the Nernst equation here. And the voltage, if I take an electron out of ferrocytochrome C, which is the protein upstream from the cytochrome C oxidase in the respiratory chain, we can see that this process here has a positive 2.6 volts. In other words, if it's positive, it can happen. If it's negative, it can't happen. Remember that? Okay, good. A lot of nodding and not so nodding. That's good. But then if you look at superoxide to oxygen, that actually is a downhill reaction, plus 3,3. And if you add those two together, that's the overall chemical electron potential. And why do we want to think about that? Well, we want to think about that is to find out, you can read a lot of things in the literature where somebody will say, oh, well, the electron just simply goes away in biological systems. Well, no, there's a rhyme and reason why we have electrons going where they want to go. And it's dictated largely by this type of an equation. So from here, we can actually use the ferrocytochrome C, because this Sorey band here, you can measure superoxide. It's a classic way of measuring superoxide by superoxide reduction of ferrocytochrome C to ferrocytochrome C. You get a Sorey shift, and it's a really nice and easy way to quantify superoxide, even on cells. So if you think you have ROS on the outside of the cell, you put this on there, you take a spectrophotometer, you measure the difference, and it works. So let's go back. There's a pecking order. I like to call this a pecking order for all these different species. So this is a really nice table. It was produced by a group in Iowa here. And this is your classic hydroxyl radical here. Sorry about that, it didn't translate here. But these are your different radicals. Hydroxyl radical is very powerful, 2.3 electron volts. It wants an electron. It'll take it from anything. So when we have radiation, for instance, it hits water, you generate hydroxyl radical and a hydrogen atom. that hydroxyl radical will attack almost anything in the neighborhood, almost diffusion control. Aliphatic, these are from lipid radicals. Glutathione, we'll talk about that in the second lecture of the biochemical series. And glutathione is a very powerful antioxidant. We also have hydrogen peroxide going to hydroxyl radical, but that's still uphill. Ascorbic acid, again, it's milder, so these can take electrons from this systems here, and then we have superoxide being converted over. It's uphill by 0.36. Paraquat, of course, the iron cysteines, and of course, reduction of water is actually a very uphill battle here. So the reason we have this table here is to kind of give you a feel of who is the most powerful oxidant, who's the most powerful reductant. So what is the chemistry? So electron transfer, I guess I would say my scientific grandfather was Henry Tauby. So I was part of Henry Tauby, who was the one who brilliantly described electron transfer for the first time in the 1970s. And the way that he described, there are two, outer sphere versus inner sphere. So this is oxidation. You have a substrate, and you take out an electron, that's oxidation. If you put an electron in, that's reduction. But there is something called outer sphere and inner sphere electron transfer. If you've ever seen one of those Van de Graaff spark generators, where the electron goes from you, you get a nice spark or lightning, that's outer sphere electron transfer. You have an electron here, and it hops over to the other molecule. But inner sphere is a little different. Inner sphere is where you actually have to form a bond first, and then the electron goes from one side to the other. And Mother Nature likes to do that more than outer sphere electron transfer. And the reason is that you don't want free electrons floating around biological systems, because they might get usurped by something else. So this is a very important concept here, outer sphere versus inner sphere electron transfer, because it tells us how the electrons are going back and forth. So it's interesting, everybody talks about outer sphere electron, even in the respiratory chain of the mitochondria. But if we look at the mitochondria, it's brilliant, because it's actually more like a wire. It's more like a solid state problem, where the proteins are here, and they're juxtaposed to the other one, allowing this outer sphere electron transfer, but it's kind of insulated, so it doesn't go anywhere else. It's an insulated wire. So Mother Nature's really smart about this. So here's examples of outer sphere electron transfer. The most simple and obvious one is oxygen by electron makes superoxide. We all hear of superoxide all the time. And superoxide by itself doesn't stick around. I mean, people have tried to estimate its concentration in the cell, and it's about femtomolar. The reason is because it rapidly dismutates. In other words, two of these react into water and hydrogen peroxide. Oxygen can also pick up two electrons and form hydrogen peroxide. But if you take an electron out of hydrogen peroxide, you generate the hydroxyl radical, which is a powerful oxidant. If you actually take two electrons out, you'll get H2O and oxygen. That's the catalytic mechanism. If you put two electrons in, you actually get H2O. So you have ways of pushing and pulling these species here by just simple outer sphere electron transfer. Here's examples, though, on inner sphere electron transfer. A lot of people, if you go through the literature now, one of the most popular signaling pathways, of course, is through thiols and the oxidation reduction of thiols. So if we look at thiol reduction, if you have a double disulfide bond here, if you take DTT, which we all have done, and we reduce the thiol here, that actually has to go initially through a bond. In other words, the thiol, the DTT actually binds to one sulfur, gets oxidized, and the two sulfurs go away. But you have to form a bond first. Disulfides, this is classic. Oh, I'm sorry. This is again classic. You have a disulfide here. If an oxidation occurs here, you form this disulfide bond here. Hydride transfer is another very interesting. You notice NADPH, NADH, FAD. It's a great way to transfer electrons through hydrogen atom or hydride transfer. If you put a hydride on something, it's actually like a 2-electron reduction. So let's talk about oxidation of biological membranes. or biological materials, I should say. This is bad. This is always considered deleterious. And so the hydroxyl radical in this case here will abstract, because remember we said the hydroxyl radical was very powerful and it could abstract a hydrogen atom here and form an alkyl radical here. And that, like ribose, it can actually oxidize ribose here. And we'll talk a little bit later about some of the ramifications of this chemistry. Hydroxylation. The hydroxy radical here, which is the most powerful of all the oxidants, can actually hydroxylate material. It can hydroxylate things like guanine to form 8-oxo-DG, and some of these have been markers for oxidative stress. There's other ways of these powerful oxidants, like these metal oxo species, which are almost as powerful as a hydroxyl radical, and they can actually insert into a nucleophile and oxidize different nucleophiles. So this would be an insertion reaction. So there's different mechanisms of how different oxidants behave. And the metal oxo species actually play a larger role in biological systems than just a free hydroxyl radical. Those are really under very special conditions. So these are the nitrogen oxide related terms. There is something called nitrosylation. Now nitrosylation and nitrisation in the literature often get mixed up. I'll point that out to you. From a chemist's point of view, nitrosylation is where a substrate reacts directly with NO to form an S-nitrosyl complex. This is metals and radicals. Often you'll see in the literature S-nitrosylation. Technically, that is wrong. The reason it was coined a lot of times was that it kind of reminds people of phosphorylation. So there's been a kind of a nomenclature argument, the chemists versus some of the biologists, whether you can use nitrosylation. But from a strict chemical point of view, nitrosylation is the reaction of NO with a substrate. Nitrisation is the transfer of an NO plus moiety to a nucleophile to form a nitroso species. Classic example, and we'll talk a little, is that thiols react with N2O3. It's formed from the things like the autooxidation reaction to form SNOs, or amines to form nitrosamines. The other term that's very important in nitrogen oxide-related chemistry is nitration, where nucleophiles plus NO plus can form nitro groups here. And the most common and one of the key footprints is the formation of nitrotyrosine. You can buy antibodies to this, and this we look as myself personally as a footprint of a reactive nitrogen oxide species. So let's go back to the chemical reactions of hydrogen peroxide. Is there any questions? If there's no question, interrupt me if you have any questions. It's fine. Or we'll just go through this, get this over. It's fine. Either way, it's good. So, okay, this is the classics, right? So this is where it all started with Britton Chance way back in the 1950s, Henry Taube, where they were looking at an old reaction of ascorbic acid, iron, oxidized different organic substrates that they noticed in the 1800s. Well, this reaction was debated a long time, and later on it was suggested that this reaction was occurring in vivo and that this was responsibility for cancer, aging, and all the diseases associated, making these free radicals, which then cause cellular death. So if we have iron 2 plus peroxide, not necessarily iron 3, you generate either hydroxyl radical and iron 3, or an iron 4 or ferryl complex here. Either one of those are very powerful oxidants, and they can oxidize DNA and lipids and some of the other things. It turns out is that whatever the ligands that are around this species here, whether it's EDTA or something like a porphyrin and sulfur, determines whether or not it has more hydroxyl radical production or more iron oxo production. And that's really important because hydroxyl radicals tend to be a little more powerful than the iron oxos. The iron oxo species also tend to be much more specific. And oh, by the way, these can be copper, nickel, cobalt, manganese. A lot of these metals will react with peroxide. So heme complexes. This is like your hemoglobin, right? So the chemistry of hemes. So you have redox, as Terry was talking about, a lot of molecules, these redox molecules react with heme proteins. In fact, the reactions are generally almost diffusion controlled in many cases. Oxygen will react with ferrous iron, CO, hydrogen peroxide, even superoxide, NO, and HNO. All these will bind to a heme center. And that has different ramifications. In the case of hemoglobin, myoglobin, of course, oxygen, CO can bind and block that, as Terry talked about earlier. The peroxidases, we'll talk about that in the second lecture. I think that's in 2 weeks, about the chemistry of the peroxidases, which is very important in handling hydrogen peroxide. Cysteine, if it's cysteine in the acyl ligand here, is for the P450s and nitric oxide synthase. And by changing this acyl ligand, you actually change the different properties of this iron here. And tyrosine here is catalase, which actually is major consumptive pathway for hydrogen peroxide in the cell. So peroxidase activity. So basically the chemistry of this is where we have an iron 3 complex. This would be myeloperoxidase, things that are secreted by the neutrophils. And they'll secrete them out. And the reason that they want to do that is they want to generate these reactive species to essentially sterilize the environment. So these react and form what they call compound 1, which is this iron 5 porphyrin, or the iron 4 pication radical more correctly. That's a chemical term where the radical is really out on the porphyrin rather than all on the metal center. And so then our substrate can reduce this to compound 2, which is an iron 4, and that can return back to another substrate oxidation to the native state iron 2. This iron 2 can be reduced to iron 3 can be reduced to iron 2, substrate oxidize that to iron 2, and then back to iron 3. So there's several ways that we can do this. We can either get reduced here to the iron 2, go this way through the iron 4, or we can form the iron pication radical to iron 4 back into the substrate. And myeloperoxidase and the halobromo hydroperoxidase, these are classic examples of these type of peroxidases. Another thing, a substrate is nitrite, and nitrite can be actually oxidized to NO2. And why is that important? Well, controlling of pathogens like tuberculosis, and TB, NO2, we don't know why, but NO2 is a very powerful toxin to TB. And so the formation from nitrite, either through acidic nitrite, which we'll talk about a little later about, and formation of NO2 through these peroxidases can actually form an anti-pathogen response. So we go back and talk about the classic Haber-Weiss chemistry. If you have superoxide again can lose its electron or it can gain electron to get hydrogen peroxide. So in the case of the Haber-Weiss cycle here, we have iron 2 plus peroxide, and that can make hydroxyl radical. Hydroxyl radical doesn't just sit around. It finds a nearest substrate like hydrogen peroxide and regenerates superoxide. Why that's important is because superoxide can re-reduce to iron 2, and then the iron 2 can react again with hydrogen peroxide here. So you have this cycle here, interlinking cycle here, which actually then can facilitate the catalytic production of things like hydroxyl radical. And this is what we think leads to a lot of radical reactions in biological systems that get out of control, leading to cell death. Again, we mentioned dismutation early on, 2 superoxides to oxygen H2O2. This is a pH-dependent mechanism. And even though this is a fairly fast reaction, Mother Nature has actually developed enzymes. These are superoxide dismutases, which actually catalyzes this reaction to almost diffusion control. And it will, like the copper, which is copper goes to copper +1, the second superoxide comes in and oxidizes to hydrogen peroxide plus copper +2. And this is very important because there are several key SODs. The first one is copper zinc SOD. We'll talk a little bit about that again in the next lecture. That's mostly in the cytoplasm. That controls the superoxide formation in the cytoplasm. The manganese SOD is actually one of the more interesting ones because it controls the superoxide production and the electron flux inside the mitochondria. And there are a lot of interesting papers that showing them having high and low manganese SOD and controlling the mitochondria has a lot of effects on metabolism. It also has a lot of effects on cell proliferation. So it's more than just scavenging radicals. It's actually shunting electrons to different places. Superoxide can also react with the conatase. And again, this is important because the conatase being part of the TCA cycle, or Krebs cycle, can be inactivated by superoxide. By scavenging superoxide, you can prevent the Haber-Weiss cycling, so you don't have this out-of-control looping effect. And it also prevents NO scavenging. And that's actually one of the most important things about the interaction between superoxide and NO and peroxynitrite. It isn't really the formation of this, which we will talk about later on in the lecture here, but it's actually taking the electron flux for superoxide going to ROS and taking NO from its signal transduction pathways like guanylate cyclase. That's actually one of the more important things that it does. We talked about the hydroxyl radical. One of the things that's really important is how do we detect this? And this is one of the, I'll tell you, this is a really difficult thing. You'll read it in many books saying ROS dies, DCS, F. I mean, everybody uses it. But dyes just generally tell you about the oxidative state of the cell. It doesn't really tell you the nature of what's happening there. And some of the ways that people have done it is through spin traps. There's some very clever mechanisms. Kali Raman has, in Wisconsin, has done a lot of this work here. If you're interested in ESR, I'd suggest you go look at his or Murali Krishna's work. Trapping in a test tube with benzoic acid and salicylic acid, the hydroxylated products, is another way of detecting ROSs. The dyes can be complicated, and they don't always work. Everybody comes to me once in a while and says, why didn't this dye work, or why doesn't that dye work? Well, they're complicated chemistries in a complicated biological system, so you have to be a little careful, and you have to be very careful of your controls. So let's talk about DNA oxidation. We are cancer, and we're concerned about oxidation of DNA. There's the oxidation by many species, such as the ROS, that oxidation of ribose, which leads to strand breaks, oxidation of the bases, which can lead to base oxidation, cross-reaction, and deamination. If we look at the proteins, where the oxidative damage of the proteins. This is methionine. Terry had mentioned methionine. Methionine is a really good antioxidant for ROSs because it can actually bind the sulfoxide here. It can also react in a radical mechanism here to form methionine sulfide. Cysteine, if you take, one of the things about reactive species chemically is if you take glutathione out of the cell. Has anybody ever done that? Well, if you want something fun, if you're looking at your cells and you take the glutathione out with BSO, the cells are simply very unhappy because the glutathione is really critical in managing the ROS and the reactive species in the compartments of the cell. So the glutathione is really your frontline mechanism here. And when you have thiols here, and you form this radical here from things like hydroxyl radical, there's a series of reactions these thiol radicals will undergo. One is, of course, dimerization here, which can generate superoxide and RRS, or it can react with oxygen, form this peroxo species here. That can fall apart to other species here, such as the sulfoxides here, or it can form sulfinic acid here and go back to RRS. And this variety of chemistry becomes very complex. This is another reason why biological systems don't like radical chemistry. It's complicated and uncontrollable. Lipid peroxidation. This is one of the classic mechanisms of cell death. You have an initiation by iron oxide or hydroxyl radical. You form this radical here. The radical reacts very rapidly here with oxygen to form these peroxo species. The peroxo species then can abstract an hydrogen here, forming another radical here, which can react with oxygen again. But you have this hydroperoxide, which reacts with metal to form these alkoxide radicals, which can come back and start oxidizing these again. And so one oxidant up here can lead to a series of lipid oxidations, numerous propagation of lipid peroxidation. And this is one of the ways they think, which has been proposed, of cell death with ROS. This is just sort of examples. I put this here of good and bad lipid peroxidation. Here are membranes, and you can, in the membrane, you can actually perpetuate these type of lipid oxidations and destroy the membranes. However, lipid oxidation is not always a bad thing. The P450s and the cyclooxygenates, the reason we take Advil or I take diclofenac, is to actually prevent all these products here. And the prostaglandins and leukotrienes, which are actually very powerful signaling agents, both in the cardiovascular and the immune system, actually rely on lipid peroxidation. But the difference is that those enzymes control the lipid peroxidation within the confines of the protein. Where when we have the membrane, it's not. It's just sort of running down and destroying the membrane in a cyclic manner. This is tyrosine oxidation here. A lot of times you get tyrosine products. In the old days, it was looking for tyrosine radicals here. You can form nitrotyrosine, or you can hydroxylate this. Many proteins get modified this. Proline plus iron oxo species. This is the HIF. This is what Terry was describing. The chemistry is the prolohydroxylases are key, and from cancer people, we're used to talking about in terms of HIF, or IKK beta. So it regulates also the NF-kappa B system. But if you go back, you need ascorbic acid. And we all remember what scurvy is, right? If you don't have enough vitamin C, you have scurvy. And scurvy, this enzyme here is also critical in hardening the collagen, increasing its melting point. So this enzyme here, even though it's doing sort of the same oxidation, has very key functions, both in signal transduction and also the health of the cell and tissue. Earl Statman founded this, the famous Earl Statman, who was in this building and also Building 3. The Statman, Statman Fellows are actually named after him. And I actually knew him, he was a very famous oxidative stress guy. But he actually showed these carbonyl formations in proteins as evidence that there's oxidative stress in the cell. Sulfur oxidation, I think we've done that. Oh wait, am I going backwards? Or we have double species. Now we come to my favorite. I could tell you stories about this. This is actually more fun. But anyways, nitric oxide. Nitric oxide, I will say, we started studying this when it first came out in 1989. I know, God, that sounds so ancient. The first time we started, it was so brand new. Now it's so ancient history. They gave a Nobel Prize out for discovering why nitric oxide, this radical species, actually regulates your blood vessels. So Lou Ignarro, Bob Furchgott, oh my God, Fred Murad, sorry, I can see his face. Fred, yes. They got the Nobel Prize in 1998, and they got it because nitric oxide here has a lot of chemistry. We'll talk a little bit about the chemistry, bound to this heme called guanylate cyclase. Fred showed that initially, which was kind of curious, and Bob Furchgott had noticed that there was a substance they couldn't identify that went between the endothelial cells and the vascular smooth muscle cells. And he couldn't figure it out. Lou Ignarro, when very astutely was studying soluble guanylate cyclase, said, I bet you it's NO. So he actually did some experiments showing that it was actually NO, and he actually detected NO for the first time. Salvador Moncada also came in and showed the same thing. And this field is actually now, I think there's 170,000 citations, or 170,000 papers in this field. ROS is about 200,000. So if you just take those two fields and you compare that to the kinome, right, or phosphorylation, which has about 300,000, this is a much bigger field. So So what is the redox chemistry? Let's talk about this simple molecule. We all studied this in freshman chemistry. We all had to draw the diagram where the radical goes. I think we did. You guys still do that? I don't know. Freshman chemistry, they don't teach the same as they used to. But anyways, NO is a free radical. So again, it can lose an electron to get oxidized to nitrosonium ion, but that's a lot of energy. It takes a volt to do it. And you can reduce it. But nitric oxide takes a lot to reduce this. And this nitroxyl here, and we'll talk a little bit about this, that means it's very hard in mammalian biological systems, not necessarily bacterial, but mammalian systems, for these reactions to happen. So nitric oxide, the chemistry of it, one of its primary functions, because we divide this in how fast reactions go. So you have this small little molecule, we can buy it in a bottle, but it does go away. It reacts with oxygen. We'll talk about that in a little bit. But if you put it in a biological system, it reacts quite fast. So where does it go? Well, the biggest targets are these metals. So the NO can react with metals, such as hemoglobin, but more importantly, guanylate cyclase, because every tissue in your body has cyclic GMP, and through the NO cyclic GMP. NO can react with radicals on occasion to abate things like lipid peroxidation, so it actually has protective effects. So that's what we call radical-radical coupling. The autooxidation, which we started investigating, it's a reaction of NO to nitrite. So this is, if you put NO in solution, it's not very stable, right? Put a lot of it in solution, as we'll talk about, goes away in a few seconds. Actually, it's interesting, which we'll talk about later. You put a micromolar, it stays around for 30 minutes. So yeah, that's a really strange reaction. This is your classic reaction. A lot of this was studied, a lot of this chemistry was studied by PChem people. LA pollution, where Terry was. You remember all the, that kind of, you guys, they cleaned it up quite a bit. But in the old days, in the 70s, I remember getting first to California, right? And my friend takes me down to LA. I was in Santa Barbara. We take to LA, and I wake up in the morning, and I swear to God, I was in New York, right? And you see this like monster coming down the valley. It's brown gunk just coming at you like this. And it was literally like a brown fog back then. And some days you couldn't see your neighbor's house. The NO2 and all this was ozone was so bad. Kind of smelled like ozone too. But that's where a lot of this chemistry got its start, was trying to understand air pollution. So this reaction, though, has a lot of unique kinetics. And we were really surprised because we couldn't explain a lot of its behavior. Because you have this paradox in biological systems with NO. You've got something that is so toxic that it comes down and looks like it's going to kill all mammalian life in LA. It doesn't, but it looks like that, right? And then you have this phenomenal thing. If you take NO away from you in your cardiovasculature, all of a sudden you have every chronic disease under the planet. So it's really important. So how do we understand this dichotomy between NO being unstable in aerobic areas and NO actually being very important in cardiovascular and other cell components? Well, it turns out the basic fundamental chemistry, and in 1970 something, I was taking freshman chemistry. I won't tell you exactly how old I am, but my teacher had actually told me, I'll never forget this day. We're studying third order reactions, and she said, this reaction will never be understood. I can say in 2016, with the help of another student 3 years ago, who's a professor at SMU, we've actually understand it now for the first time after 50 years. But in this case here, this third order reaction here was unique, because what this said is that when you're at 100 micromolar in the presence of oxygen, the lifetime is 8 seconds. But because it has a second order dependency, as I dilute the nitric oxide down 1 micromolar, the half-life increases 800 seconds. So the physiological concentration, as we know, is in the nanomolar range. So if you make NO in an endothelial cell, it won't react with oxygen for an hour, which, as we know, all these other proteins are sitting there. So at low levels, nitric oxide is a wonderful signaling agent. You see what I mean? But now I'm a macrophage. Now, the other part of the NO story was John Hibbs and Steve Tannenbaum, who said, oh, there's this nitric oxide-like species making nitrosamines and killing bacteria and killing tumors. It turns out that the other part of the NO story is not only regulatory, but the NO story is also pathogenic and anti-tumor. And so when you go to the higher concentrations here, let's say we get it up now 1,000-fold more, maybe 10,000-fold. All of a sudden, those species go up astronomically, and that becomes a predominant reaction. So we kind of tried to sort all this out. And in 1998, I was working over in Building 10, which I worked there until just last year. And you talk to a lot of people, physicians and other people, and they said, we really don't want to understand the chemistry. One of my first bosses here, Jerry Rice, said, I gave my first talk coming from MIT. We talked in differential equations, kinetics, and he said, you're not allowed to give more than one differential equation per talk. So then, my first NIH thing, I couldn't talk PChem, right? So anyways, so I kind of made up this, how do we explain this on a qualitative way? And so basically we looked at the NO, chemical biology of NO, and direct effects and indirect effects. Direct effects were the chemical reactions that are fast enough where NO can bind to them, heme proteins. Not sulfurs, but heme proteins. Also other radicals. And this explains a lot, and it reacts with things like oxyhemoglobin very fast. So NO can't just simply diffuse around the body because the hemoglobin actually is scavenged like a black hole for NO. It can regulate everything inside the tissue, but once it gets into the blood vessel, it's gone. The guanylate cyclase. But if we get in an area where we generate, like in a macrophage, a mirroring macrophage, humans are different, but we haven't quite figured out the difference between humans and mirroring systems. But in a mirroring macrophage, we know we generate levels that are above, fluxes above 0.5 micromolar for days. And we do generate a lot of these species here, and including nitrite here, which can be reoxidized to NO2. And these are what we call the indirect effects. And the reason we call them indirect is because they're not being mediated directly by nitric oxide, but in fact by these reactive species here themselves. So I think we had double slides here. Deja vu. Yeah. Sorry, my bad. So what are the direct reactions? Again, we'll go over them. Nitrosylation. This is real nitrosylation, where NO reacts with the iron complex, forms an iron oxo. Of course, the best example of this is soluble guanylate cyclase. NO can also react with hemoglobin. It makes methemoglobin nitrate. This is a very good way of controlling the nitric oxide in vivo, but it's also a great way to detect it. In fact, as Lou Ignarro, to discover NO, why he was given the Nobel Prize, is he cleverly used this reaction here to actually detect it in endothelial cells. Very clever. Also, NO can react. Remember we were talking about those reactive oxygen species and how powerful they can go and oxidize different species? Well, NO actually reacts with them and abates it. So NO antagonizes all ROS. They seem to antagonize each other. So they actually chemically form this. And I can actually, just in the homeostasis of your blood vessel, right? You have, if NO goes up, you get relaxation, right? But then you get constriction with the calcium influx. ROS goes up. That scavenges all the NO. The calcium influx causes constriction. The NO comes back up. It causes relaxation through scavenging of NO and ROS. But if you think about that, if you're in the heart and you're out there running, I can't run anymore. I cycle instead. Right, Terry? We have bad knees. You want that moment to moment, that second to second, that millisecond to millisecond actual translation of your blood pressure, right? Into different vessels. Neuro, same thing. And one of the ways you can do this is the balance between NO and ROS. When you get imbalances in that, that's when things kind of go out of whack. Atherosclerosis is this chronic favoring of ROS, and then it recruits leukocytes to the arteries. So Haber-Weiss chemistry. So this is a really important thing, I think. And this can apply to any of the peroxidases. If you have iron plus peroxide, we form, again, this hydroxyl radical. NO can react at diffusion control to form nitrite, which abates this reaction here. You can form superoxide here. and the superoxide, NO can react with it to form peroxynitrite. And this was very controversial a long time ago. We'll go over this later. The virtue of forming peroxynitrite in our hands and many other people's hands actually is an antioxidant because it's preventing the more powerful oxidants from being generated. It itself is a powerful oxidant, but as we'll talk about later, this is rapidly scavenged also by NO to form nitrates and nitrites. So this is a very powerful antioxidant property. Lipid peroxidation, remember we were saying that lipids can form these oxolipids, lipids with NO, and NO can actually form these peroxonitro complexes here. Bruce Freeman and co-workers have actually said these nitrogen-modified lipids actually have positive health benefits. Bruce Freeman shown some really powerful effects of nitrolipids. The fact is that he says a lot of the fish oil you get has nitrolipids in it, and that's actually the active ingredient, it's the healthy part. So if you actually form any of these lipid radicals, the reaction with Sarah Goldstein actually measured these a long time ago, but these are almost 10 to the 9th. They're very rapid reactions, and they terminate the lipid peroxidation. So what about the indirect reactions where NO can form N2O3, NO2, peroxynitrite? We can result in nitration and lipid oxidation. And so how does this balance happen? Well, nitrosation chemistry. The classic one is N2O3 here. If you remember from your freshman chemistry here, this is your N2O3. The nucleophile will bind to it. And one of the classic is that N2O3 combined to form S-nitrosothio complexes here in nitrate. Nitrosamine formation can form these secondary nitrosamines, and these secondary nitrosamines are important. In the 1970s, this reaction was happening in industry, in the beer industry, and they were forming nitrosamines. And nitrosamines are actually notoriously carcinogenic, at least in rodent models. And so it shut down the German beer industry. This was a national crisis because now they found this carcinogen in processing of their hops. But they actually figured it out, and within 6 months, the beer industry was back producing it with nitrosamine-free. So the nitrosamines are actually can be very powerful liver carcinogens. Another thing is that N2O3 can react with primary amines, such as those in DNA, like guanine, and they can cause deamination here. And this deamination process here can lead to some types of mutations. Nitrosation targets, protein targets. You can S-nitrosylate many different proteins here, such as GAPDH, HIF, NF-kappa B, a variety. But in our pathway, we've been working on nitric oxide. We found that NOS2 actually is a very powerful predictor of poor prognosis for ER-negative breast cancer. And we just did a forum on NOS2. NOS2 produces the higher levels of nitric oxide. And what we found in humans was that when the cancer cell produces the NOS2, the prognosis is very bad. In fact, it can be as bad as hazard ratios of 6 in 5 years for ER-negative breast cancer patients. It's also in triple-negative breast cancer. Jenny Chang has discovered the same thing, and is in fact looking at this as a clinical trial that they're going to start for NOS inhibitors. But the way it works, we think, is nitrosation of key proteins like EGRF, SARC, and RAS. Even though we can mutate RAS, everybody talks about mutations of RAS, what we find is that's just like turning the key of your car on. That doesn't fully activate RAS. There's a critical thiol in RAS that when it gets oxidized or when it gets nitrosated, it turns it on steroids. And it actually promotes a variety of different pathways. In fact, is the chemistry of nitric oxide, from an oncogenic point of view, is if you look at the Weinberg map of all the properties, PI3 kinase pathways, HIF pathways, KLF pathways, nitric oxide activates not just one, but it activates all of them simultaneously. And this is why it actually, we think, is promoting cancer. In this case here, also NO can react with zinc fingers here. PARP, it can inhibit PARP and these different DNA repair proteins here. And tyrosine, nitric oxide can actually form nitrotyrosine. We showed this as an important signaling pathway, actually in ER-negative breast cancer. It can react with lysines to cause a deamination. And this happens with ligase here. I'm sorry. And it can react with tryptophan, but I don't have any examples of any modification of protein activity. Again, NO2 can react with thiols. We talked about the thiol radicals, and it can generate these thiol radicals here, which can actually generate a number of different radicals here. And because you can get into NO2 from the NO-oxygen reaction, acidic nitrite reaction, or peroxynitrite reaction, the thiols actually become a very important mechanism of controlling the RNS. Oxidation and nitration of tyrosines, the same thing. You can form NO2. NO2 can form this radical here, resulting in tyrosine dimers, which are detected often in vivo, or nitrotyrosine as well. Nitrosative mechanisms can get you to the same place for the nitrotyrosine. N2O3 can form this complex here, which then can rearrange to nitrotyrosine. The summation of the indirect effects, therefore, are NO can generate NO2, N2O3, and form nitrosation nitrate. But NO2 can result in nitration and oxidation of proteins. You can form peroxynitrite, but peroxynitrite reacts very rapidly with NO, not directly, but through its reactive form to form NO2. And the other thing is, because we have phagosomes, autophagosomes, those are acidic environments. When you have acidic nitrite, you can actually generate a lot of this chemistry just from acidic nitrite. In fact, the phagosome is one of the places where the acidic nitrite is actually very important in controlling pathogens. So I'll just sort of leave you with what we're actually working on, nitric oxide. So this is from one of our reviews here. And I won't go into this too much. We don't have enough time. But basically, we talk about NO chemistry, right? But the levels of nitric oxide, so we asked the question, we had the ability to do this. NO can be good and bad. You've always had this dichotomy in any disease. So we decided, well, maybe it's just as simple as concentration. A good friend of mine, Glaxo Welcome, we were sitting on a beach somewhere at a conference, and he said, I said, what's the difference? He goes, oh, it's the amount of NO, I think. So we started looking at this a while ago and trying to piece together why NO might actually promote cancer while being deleterious. Well, what we found is at this 10 nanomolar level, which is basically regulating cardiovascular disease, neuro, you go mostly through cyclic GMP. And we've shown that thrombospondin 1 controls the cyclic GMP. Dave Roberts, who you'll hear talk later on in the session, has a beautiful story on how thrombospondin is the antagonist of cyclic GMP and through CD47. But when you cross this threshold and you start getting up in 50, 100, 300, you start activating other systems that are cyclic GMP independent. This is activating an AKT system. You activate HIF, which results, as Terry showed you, VEGF. And you also activate SRC, EGFR, RAS. And these pathways then subsequently act things like beta-catenin and NF-kappa B. And you drive this more mesenchymal or more cancer stem cell-like phenotype in this area between about 500 nanomolar and about 100 nanomolar. And this is where we're finding in cancer cells that express nitric oxide synthase 2, we increase cancer metastasis and cancer cells send this phenotypes. However, what was surprising is when we went over this, we went up to 100 nanomolar flux, or 1,000 nanomolar fluxes. We studied this with Kurt Harris, Pervez Hussein, who we'll talk later on, and Kurt Harris, and who, what we were able to show here was we get phosphorylation of P53, and in this range here, we actually get nitrosative stress responses. So the levels of NO can actually promote, or it can increase stress, or it maintains homeostasis. So just based on the concentration, and the concentration dictates what the kinetics are of each of one of those targets, what the chemistry is going to be. Actually, the cells have sorted out into this distinct signaling pathway. And so above here, at this really high dose, we get all that crazy chemistry with ubiquitation of nitrosation, nitrosative stress. We activate things like P53. But below that, we find that nitrosation, nitric oxide reactions in the membranes are actually promoting a lot of the same things that growth factors do. And then below this, we have the cyclic GMP pathway, which is really critical in homeostasis. So that's all I have to say. I hope we got through that fast enough for everybody, the chemistry. But that's, if you have any questions, just email me. And this is sort of kind of give you the basic language of redox chemistry. And so when you read something, you kind of go, that's what an oxidation is. So thank you.
Any questions today?
Now they want to get out of here. It's time to go. But you're going to sit on 270.
You were.
I'm going to sit on 270.
But you were.
Thanks.